afternoon, everybody. My name is Sam Srivastava. I'm the chairman and CEO of Venn Therapeutics. We are an immuno-oncology company. And after I sold my first company in June of 2015 to a NASDAQ-traded company, my second company was Venn Therapeutics, and I started with a big idea on the back of an envelope. The big idea was very simple, that there's an adaptive compartment of the immune system, an innate compartment. We wanted to really prime the immune system. As you've heard from a lot of people, that you would want to take the cold tumors and turn them hot. So we were about 15 or 18 months ago, that was our idea, to make sure that we are building a company, a pipeline that did just that would be symbiotic with checkpoint inhibitors. We had no desire to play in the very crowded and competitive checkpoint space. So we started out, first thing you have to do when you build a great company from the foundation is to have great people. So I want to recognize my scientific advisory board. We were very fortunate to have Dr. Edith Jansen from Cincinnati Children's Hospital. She's an expert in looking at the innate to adaptive pathway. She had done her prior work with Dr. Bruce Butler, who won the Nobel Prize in 2011. We have also had Dr. Daryl Irvine from MIT, Howard Hughes investigator at the Koch Institute, and Dr. Paul Reinert. He's a very highly respected immuno-oncologist consultant. So we are a privately held company, and we are very fortunate to attract such high caliber of talent. What I want to share with you today is our pipeline. So our pipeline is really focused on having the best-in-class ideas for immunopriming. And you know what? 16 to 18 months later from when we started, the field is coming to us now. We are very excited in the advancement of our pipeline, but also the field in general. Because at the end of the day, it's about the patients. And the patients are going to be the recipient of the benefits here. So let me give you a very basic example of a cold tumor. As you know, you can take the brakes off the car, but if the car engine is not turned on, it's not going anywhere. So you have to turn the car on for it to go somewhere. And that's why you see 70 to 90% of the patients now respond to PD-1 therapies or PD-L1 therapies because the engine is turned off or the engine is stalled. So I'll show you today how our assets either turn the engine on or take a stalled engine and make it work. So they're immunologically cold tumors and we make them hot. Our lead asset is a STING agonist, and I want to talk to you, more about our STING agonist asset. The idea of a STING agonist became front and center in March of 2015 when Aduro, a small biotech company, signed a pretty significant collaborative agreement with Novartis. The cyclic dinucleotides here, as you can see in this picture, STING is the critical mediator of spontaneous anti-tumor response. So this is a key discovery, that's happened in the last 8 to 10 years. It now is being applied to immuno-oncology, and we are very excited about it. We are actually hoping, and the response in humans that translate, because a lot of the work done so far has been in animal studies. The human studies that started last year by Aduro and Novartis collaboration. So we wish them the best because we really want to see this pathway work because it helps the patient and it helps the field advance forward. Our insight was a unique insight into the STING targeting. So if you look at the bullet point number 3 here, our inventors at Michigan State University, Dr. Andrea Malfitano and Dr. Chris Waters, their insight was to use an adeno delivery. So this is not gene therapy, but this is using the gene delivery method of delivering an enzyme called diguanylate cyclase into the cell, where it synthesizes the cyclic di-GMP, which then activates the STING pathway all the way to interferon, type 1 interferon. And on this slide, we show how potent that approach is. Look at the slide here. If I was to put a number there, the ad null, which is the ad vector itself, and our drug, that's 1,500 times. Let me repeat that, 1,500 times more potent in the type 1 interferon, or this is interferon beta expression. Our approach, we think, is going to be a potential best in class because the manufacturing is scalable. The safety profile is extremely well known, and it overcomes a lot of these cellular entry barriers that other approaches have. We were very excited earlier this year to be invited to an oral presentation at American Association of Cancer Research in Washington, D.C., in which a lot of big names presented in the innate immuno-oncology presentation. And we showed for the first time that our profile of efficacy is very strong, but our safety profile is even more exciting for us, because as a combination-focused company, that's where the name Venn Therapeutics comes from, is Venn diagram, is combine different modalities. You want to be very careful with toxicity, because you start adding drugs on drugs, toxicity becomes a real issue. So safety profile was extremely important to us. So look on the slide, the couple of key takeaways. Safety looks absolutely fantastic. Look at the expression on in the liver. So no off-target toxicity, but also importantly in tumor, if you look at the bottom, our STING expression lasts up to 48 hours. So we have the ability to titrate up or down our vector depending on how much we need to have an efficacious response. So we have two major advantages here, because typically I've been told that small molecules would have an expression for 4 to 6 hours half-life. So this is definitely a competitive advantage for us. Our efficacy looks very, very solid. This is just one injection. So let me repeat that. We only gave one injection of our drug and 2 of the adeno, just to keep things clear for us, because one injection is what it took in this B16 melanoma model to produce a very strong anti-tumor response. Our survival also correlated beautifully with our anti-tumor response. As you can see in the red line, the PD-1 inhibitor response at 53%. STING alone, our vector was 69, and our combination with PD-1 was 83%. So the p-value obviously is pretty darn good there. However, our approach was to be symbiotic with PD-1 and checkpoints. So we are excited to have this asset move into clinic rapidly. We are advancing, doing a lot of preclinical work and tox work in the next few months. And on this slide, we capture why our approach could be best-in-class potential, because we overcome the disadvantages of a small molecule. We have a wide therapeutic window. We can really have a very high level of STING activation. And our platform can be added. There are more things that can be added to the vector that we are using currently. For maybe the next time that we are looking at this for next generation, we can add more things to it. But we can, so the platform is very flexible. So we are very excited about our STING asset. I'll move on to our second asset, which is a rationally designed small molecule inhibitor of beta-catenin. So beta-catenin is a very well-known target. And if you can look at the slide, when there's overexpression of beta-catenin, recent work has shown that the chemokine, the critical chemokine CCL4, is inhibited from the dendritic cell, which is the CD103 and CD8 alpha, and so the spontaneous response is missing, as opposed to the bottom, as opposed to the top slide here that shows the absence of beta-catenin. So we think this is a classic. This is a classic case of cold tumor in which there's an immunosuppression phenotype, and if you look at the pathway, if you can reverse the phenotype of immunosuppression here, this becomes an important modality for turning a cold tumor hot. So our approach is in the nucleus. We are nuclear beta-catenin. It's a very potent and highly specific small molecule, so it's orally bioavailable drug. And other approaches, as you have seen in the past, have been more upstream in the pathway, and this is very much downstream. There's a simple rule of thumb that I was taught, that if you're trying to be developing an agonist, you want to go as upstream as possible, and when you want to antagonize something, you want to go as downstream as possible. In this case, our beta-catenin BCL inhibition really goes down in the nucleus, so it's a nuclear beta-catenin A, and B, it only impacts the oncogenic functions of beta-catenin. It leaves the other functions alone, and we have seen that with our data, and we are frankly very excited about this asset. Our next step here is to continue to do the work here, some more preclinical development, and take this into clinic in the next 12 to 15 months. Our last asset is a oncolytic virus. As you know that the TRex has been an approved oncolytic virus drug. Our advantage here is very simple, is ours is a non-human virus. So it's a bovine, it's a cow-based virus that does not infect normal or does not replicate in normal cells. And so there's a long history of this of this virus that it has no or lack of pre-existing immunity in humans. So we think that gives us an opportunity to give it systemically. It does not cause disease in humans, and the breadth of infectivity so far does not depend on any specific tumor type. So as you can see on the slide, the bovine herpes virus is a family, is part of the HSV family. It has all the advantages of HSV, and we are very thankful for TRex to be approved and be offered as an opportunity for the patient as another option. But BHV can be the next generation oncolytic virus because it can probably reach the metastatic tumors. We can give it systemically. The potency looks very solid here, and we're about a few months away from an IP-ready engineered vector. We're adding a few more things to the wild type to the wild type virus. And frankly, we're very excited to work with Dr. Karen Mossman at McMaster University in Canada. She has published a lot of data on this specific wild type virus that is available in the literature, and the data looks very promising. So here are some of the key catalysts that are coming up for us, if you want to take a look at them for a few minutes. We have a lot of catalysts coming up for the next 18 months. Our company is looking to raise about $8 to $12 million in a Series A. I funded this company from the inception with the proceeds from my first sale, from my first successful exit. We have taken some friends and family money. So we are now out in the marketplace to look for partnership opportunities with pharmaceutical companies, and we have significant interest there, and also raise a Series A in conjunction with that. So with that, I just want to share with you that we're excited at Venn Therapeutics to develop this immuno-oncology portfolio of turning cold tumors hot and be either symbiotic or be a monotherapy as we advance our assets forward into clinic. And we appreciate your time very much, and thank you for your attention. I'm happy to take any questions at this point.